Complete dominance. If F1 is identical to one of the parents for a particular character, it is called complete dominance. For examples, plant height in pea plant. If tall is crossed with dwarf, then the F1 generation will be tall. So dominant allele shows complete dominance. Seed shape in pea, first studied by Mendel. If round is crossed with wrinkled, F1 will be round. Incomplete dominance or partial dominance. The phenotype of heterozygote F1 is intermediate between the two parents. For example, flower color in 4 o'clock plant Mirabilis chalapa. If red is crossed with white, then the F1 progeny will be pink. Partial dominance facilitates the identification of homozygous individuals in F2 itself. However, in case of complete dominance, one has to grow F3 progenies from individual F2 plant to identify if F2 is heterozygous or homozygous for a given trait. Codominance. Both the alleles of the gene express themselves in the heterozygote. As a result, heterozygotes from such genes possess the phenotypes produced by both the alleles of the gene. Example, blood group antigens in human beings, ABO blood group system. ABO blood group is controlled by the single gene IA, IB and IO. IA produces antigen A, which gives rise to blood group A. Antigen A is present on the surface of RBC. IB produces antigen B, which gives rise to blood group B. Parent IA and IB alleles are codominant, so both the alleles express simultaneously in the individual, and the blood group of child is AB. Overdominance. If the intensity of a character governed by a gene is higher in the heterozygote as compared to two homozygotes. For example, in Drosophila, small w gene governs white eye color, capital W genotype has dull red eye color, capital W small w genotype shows bright red eye color. It is due to the effect produced by the interaction of the two alleles in heterozygote. Multiple alleles. Allelomorph, another form, is the alternate form of gene. Many genes have two alternate forms, but several other genes have more than two alternate forms. 3 or more allelic forms of a gene in a population of organisms are called multiple alleles. For example, eye color in Drosophila, fur color in rabbit, ABO blood group system in human beings. These traits are governed by multiple alleles. ABO blood group in humans. It was discovered by K. Landsteiner 1900. It was found that there can be two antigens A or B, and as a result 4 blood groups. Along with antigens, antibodies are also present in serum of blood. General principle of antibody and antigen relationship is that antibodies in a particular individual will be found for those antigens which are absent in the blood of this individual. Different blood groups and the antigens and antibodies associated with them are given below. Blood group A, antigen is A and antibody is B. Compatible blood groups are A and O. Blood group B, antigen B and antibody A. Compatible blood groups are B and O. Blood group AB, antigens AB, antibody none. Compatible blood groups A, B, AB and O. Blood group O, antigen none, antibody AB and compatible blood group is O. Genetics of ABO system. ABO blood group system is controlled by single gene designated as I. The gene I has 3 alleles, IA, IB and IO. Allele IA codes for antigen A, allele IB codes for antigen B, and allele IO doesn't code for any antigen. IA and IB alleles are codominant, so individuals having IA and IB genotype have both the antigens on the surface of their RBCs and their blood group is AB. Pleiotropism. In general, one gene affects a single character, but many genes are known to affect more than one character. Such genes are known as pleiotropic genes, and the condition is termed as pleiotropy. For example, in Drosophila gene for white eye color small w also affects shape of sperm storage organs in females. Gene for vestigial wings. Notched wings also affect number of facets in bar-eyed individuals of Drosophila. Pseudoalleles. Two alleles of two or more tightly linked genes governing the same function, as a result of which they appear as multiple alleles. For example, alleles of Rh blood group system. Penetrance. One gene cannot determine the phenotype in isolation, but its effect on the phenotype is controlled by many other genes as well as by environment. So, penetrance is defined as percentage of individuals which exhibit the phenotype associated with a particular genotype. For example, if 80% individuals with capital A or capital A small a genotype express the dominant phenotype, it is 80% penetrance of allele A. Expressivity. Expressivity describes the degree or extent to which a given genotype is expressed phenotypically in a particular individual. For example, a red eye color in Drosophila. Flies may show different shapes of eye colors, suggesting variable expressions of allele capital W in capital W or capital W small w flies.